Well, thank you very much for the presentation. During this talk, I will give you a brief description of the main characteristics of electrogenic bacteria and why we want to understand the bacterial roles, specifically when they take part inside an electrogenic community inside a bioelectrochemical system. So, what are or what have the electrogenic or electroactive microorganisms? These bacteria have caught microbiologists' attention due to their unique capability of respiring insoluble extracellular electron acceptors. This metabolism is known now as dissimilatory metal reduction. And why is this? It's because these microorganisms can couple the oxidation of a chemical compound, mainly basic or simple organic compounds, to the reduction of a metal oxide. These bacteria are very diverse in nature, and today there are lots of genera that are known that can perform this task. The mostly known are Geobacter and Shewanella. These microorganisms, the task that they perform has turned these microorganisms a great technological platform, since it was demonstrated that they can also couple the oxidation of these compounds to the reduction of electrodes. So this characteristic has turned what we now know as microbial electrochemical technologies or bioelectrochemical systems into a great biotechnological platform for converting wastewaters into a renewable energy source, as I will soon describe. In addition, most of these bacteria can also couple the reduction of these compounds to other metals, such as uranium and chromium. And this has also turned or opened the possibility of developing or designing different bioremediation tools, employing also bioelectrochemical systems. So how does a bioelectrochemical system work? It's basically an electrochemical cell composed of two electrodes, an anode and a cathode, that are connected through an external electrical circuit, and oxidation and reduction reactions take part in the anode or the cathode. In a bioelectrochemical system, these reactions are catalyzed by bacteria. In the example I'm showing, which is a microbial fuel cell, bacteria grow attached to the anode, and they can oxidize organic compounds that can come from wastewaters. The oxidation of these compounds is performed by the bacteria, and bacteria take the electrons from the organic compounds and transfer them to the electrode. These electrons flow through the external circuit and are consumed in the cathode. In this example, they are consumed in the reduction of oxygen to water. But performing some modifications of the system, we can also reduce protons to hydrogen, so that we have bioelectrochemical systems called microbial electrolysis cells that are now a promising technology for the production of green hydrogen. Or we can also, as I said before, reduce uranium or chromium either electrochemically or by the action of bacteria in these cells. So we have another application of bioelectrochemical systems. There are numerous reports demonstrating and showing that electrogenic microorganisms can be recovered from diverse environments, such as sediments from freshwater or marine environments, from soil, or even, as I said before, from wastewaters. But mainly, electrogenic communities are recovered from bioelectrochemical systems electrodes rather than single species. And what is really interesting is that it was observed that the efficiency of the reactions that occur in these systems are mainly related or dependent on the members of the community. And that's why we need to understand how the members of the communities interact and what they do in the electrode. So we are working in a project trying to develop a specific bioelectrochemical system for the recovery of metals that come from polluted sediments. We are trying to do so by coupling different systems, I will not go into details of this, but what I want to tell you is that we are working with a specific polluted sediment that's in Buenos Aires, in Argentina. It's the Reconquista River, which is heavily polluted with different contaminants, including chromium. And this contamination affects a lot of people because this river goes through a very dense populated area in Buenos Aires. So, through a combination of different microbiological and electrochemical tools, we could recover from these sediments an electrogenic community. I won't go into details of this, but well, you can, I don't see if you can see this, but the community was composed of at least 6 genera of bacteria. One of them is Shewanella. Shewanella, as I said before, is one of the model electrogenic microorganisms these days, and we have lots of data about this microorganism. The results about the characteristics of this community that I want to share with you and discuss with you is one that is specifically interested for me, at least, that I'm a microbiologist, and is the particular structure of the biofilm that this community can develop. When we observe by scanning electron microscopy the electrode that we employed for the selection of these microorganisms, we found this amazing network. This is the surface of the graphite electrode, and we saw this amazing network of cables that were millimeter long, as you see the reference here, cables composed of bacteria. And what was really interesting, it was that we observed that the surface, the coverage of the electrode was not homogeneous. There were we could distinguish different biofilm areas. There was an area with a high density of cables, but with low density of scar cells distributed along the electrode. There were some other areas with a lower density of cables, but with more microcolonies of bacteria. There were other areas with also a higher density of cables, but in this case, the cables were running over a dense biofilm, a traditional biofilm, as we already know. And there were other areas where there were no cables, but there was a biofilm as the traditional biofilms that we know. We've observed different characteristics of these cables. I will show you some of them. They were not linear. They had branches, and as you see in these 2 pictures, and we also observed some structures that we call them blebs that might be the start of new branches of these cables. And what was really interesting also was that we've observed some incipient cables. Maybe these were some cells that were starting to build some cables, and we observed that the cells were connected through nanotubes, as you see in these 2 pictures. So, a lot of questions arose from these results and others that I'm not showing. And some of them are the ones that I'm going to discuss now with you. And it's, for example, is cable formation, this type of biofilm formation, induced by the electrode potential, or is also a consequence of the interaction between the different bacterial species? Which bacteria are capable of forming this structure? Because as you've seen, we had an heterogeneous biofilm with different structures. A part was cable bacteria and the other was not. But this structure was telling us some possible interactions between the bacteria, because we've seen different areas of specific types of biofilms. So different lines of evidence pointed at Shewanella as the possible cable-forming type bacteria. So we tried to address that question, if it was Shewanella or which bacteria, the one which was performing this biofilm. So we started with classical microbiology tools, trying to isolate the different species of this community. And we isolated Shewanella and also Aeromonas, and we compared their growth with the community itself. So we made them grow in different culture, such as a rich media or minimal media, with acetate as the carbon and electron donor. and organic or inorganic electron acceptors, so we had different growing capacities. And we also tried to grow them in a bioelectrochemical system and at 2 different applied potential, and compare with the community. And we observed that Aeromonas could not grow in the bioelectrochemical cell, in spite of the fact that it was taking part of the community. And Shewanella grew in the bioelectrochemical cell, but it gave a less current intensity than the community. When we observed these electrodes by electron microscopy, we observed that indeed Shewanella was the one that was forming the cables, but as you may see now in the pictures, the cables were not the same as the ones we observed with the community, and Aeromonas could not form cables. So these are the cables that we observed with Shewanella. These are 2 images showing like the incipient cable formations, bacteria starting to connect to each other. These are Aeromonas, in spite of the fact that we did not detect any current with this bacteria. It did adhere to the electrode. And so what can we conclude from these results? That at least the cable formation might be induced by the electrode potential, because when we had the control of the graphite bar without an applied potential, we did not see any cable. But since we observed different cable structures with Shewanella in the absence of the community, the interaction between the members might also induce this cable-type biofilm. Maybe it's a strategy to colonize a big area. And at least we could observe that Shewanella was capable of a cable-type biofilm formation. We do not know if any other bacteria can do that. Probably they can, but we did not see it. So thanks a lot for your attention.
Thank you.
And questions.